Hello everyone, we'll see Albert staining today. What you can see with the Albert staining? Well developed granules of volatin, means the polyphosphate, may be seen in unstained wet preparations as round refractile bodies within the bacterial cytoplasm. Means they are the granules which can store the food. When there is a favorable environment, they will disappear. So it's called as volatin. With what to stain? So these volatins can be stained with the basic dyes. Usually toluidine blue or methylene blue can be used for staining the granules, that is said to be metachromatic granular staining. What are the other methods that can be used for staining these granules, the volatins? They are the Albert Leyburn method, or even nasal staining can be used apart from Albert staining. Which bacteria can produce the volatin granules? Example is Corynebacterium diphtheria that produces granules. So this Albert staining can be otherwise called as metachromatic granular staining. So here we have some formulations which can be used for Albert staining, and there is a small difference with Albert Leyburn method, where instead of methyl green for Albert staining, we will be using the malachite green in Albert Leyburn method. So it's easy to prepare. Next is the Albert iodine formulation, that is the counter staining that can be used in the Albert staining. Procedure: we have to first make a thin film, means the suspected Corynebacterium species. If it is diphtheria, we can see the granules. If some other species, it will not have the granules. Okay. First we have to make a thin film of the colony, the organism, dry it in air, and we can fix it in the heat. We have to cover the slide with either the Albert's or Albert's Leyburn staining, then allow it to act for 3 to 5 minutes, then wash it with water and blot dry. Now cover the slide with the Albert's iodine and allow to act for 1 minute, wash it and blot dry. In the interpretation, the metachromatic granules, means the volatin granules, will appear bluish black. Okay, it takes up both toluidine blue and the malachite green in case of Albert Leyburn method. Otherwise, it will take up the toluidine blue and the methyl green in case of Albert staining. The protoplasm will appear green in color. Other organisms which do not produce the granules will appear light green in color. So this helps us to easily differentiate Corynebacterium diphtheriae from other diphtheroids, the other Corynebacterium species. Hope this post helps you to make your Albert staining procedure easy and helps you to differentiate the Corynebacterium diphtheriae. Thank you.